This video covers the Genome Editing Practical, one of the advanced training opportunities for students on our taught master's degrees. A central element of our taught MSc programs is the provision of training in cutting-edge research skills. Much of this is delivered in an autumn term module on practical and applied research skills for advanced biologists, which all students on the experimental MSc degrees take together. This module delivers research skills through seminars, online content, and practical work. As far as exciting new techniques are concerned, it doesn't get much more exciting or newer than CRISPR-mediated genome editing. Much of the BA836 content revolves around this technology, but we also make links to associated research methods from next-generation sequencing to statistics. One of the remarkable results of the CRISPR revolution has been the ability to edit genomic sequences in many different target organisms, rather than just the few organisms that could be traditionally edited. This ability has opened up many applications, for example in medicine, where we can remove mutations that cause hereditary diseases, or we use CRISPR to both study and treat cancers. Or in agriculture, where we introduce new traits into plants and animals. Or in biotechnology and synthetic biology, where we can use CRISPR to introduce new genes or entire new pathways into host cells and to edit the properties of these host cells as we want to. Or CRISPR has been suggested to be used to re-engineer the genomes of extinct organisms, and it has been used to generate completely new ways of regulating gene expression that never existed in nature before. CRISPR evolved naturally as a bacterial defense system against viruses. Bacteriophages introduce their genomic DNA into bacterial cells as part of the infection process. The CRISPR mechanism allows bacteria to flexibly target phage sequences and to cleave them, to destroy them, thereby preventing the phage from replicating and killing the cell. In CRISPR applications, this mechanism is being used to introduce DNA double-strand breaks into very specific locations in genomes, and this then stimulates the natural mechanisms for the exchange of DNA, and by combining CRISPR with the introduction of artificial DNA sequences, genes can be deleted, changed, or introduced very efficiently. One of the most interesting aspects of CRISPR is that its targeting mechanisms use nucleic acid interactions rather than protein-DNA contacts like other less efficient approaches. The motor of CRISPR is a protein, though. It's called CRISPR-associated protein 9 or Cas9. And Cas9 contains nuclease domains that can cut DNA double strands. Cas9 also binds to an RNA molecule called the trans-CRISPR or tracer RNA, and the tracer RNA interacts with a second RNA molecule called the CRISPR or cRNA. And the cRNA has the ability to base pair with the target site in a genome, thereby directing the Cas9 nuclease activity to that site in the genome. So the CRISPR RNA is specific to different cut sites in different genomes. In genome engineering applications, the tracer RNA and CRISPR RNA sequences are fused into a single guide RNA, which is easier to manipulate than the two separate molecules. In our MSc degrees, you will get hands-on experience with this important new technology in an extended practical, which is based on the removal of a genomic mutation from baker's yeast. So baker's yeast contains a multi-enzyme pathway that allows it to synthesize adenine. And some yeast strains contain a mutation in AD2 in the middle of this pathway, which inactivates that protein, the gene product, and which leads to the accumulation of the metabolite that would be normally processed by AD2, which is called AIR. Because AIR is deep red in color, these strains differ from the normal white yeast strains in that they are also deep red and they can be very easily visually identified. This RD2 mutation is caused by a single nucleotide change in the about 15 million nucleotides in the yeast genome, and it's kind of the yeast equivalent of human metabolic disorders like Tay-Sachs disease or phenylketonuria. Your task in the practical is to remove this deleterious mutation, and in preparation for this work, you will have to design guide RNAs suitable for targeting that AD2 gene, cloning these guide RNAs into expression vectors that already contain genes for the Cas9 protein, selection of successful cloning products, their introduction into yeast cells, and finally verifying that an editing event actually occurred in the target cells. For 3 successive years now, our students have successfully applied one of the most advanced molecular biology techniques that is still revolutionizing medicine and biotechnology.